Nothing new there. Last time I started talking about gel electrophoresis, and I talked about gel electrophoresis primarily from a perspective of DNA and an agarose gel. And there's another kind of gel. It's very similar. It's called a polyacrylamide gel. And the polyacrylamide gels tend to be used more for smaller molecules like proteins. And though proteins themselves are pretty big, they're pretty small in comparison to a DNA or an RNA. Okay? So when we think about those little holes or those little pores that sometimes people like to think of them, that the molecules are traveling through, okay? The agarose gel provides the biggest pores or the biggest holes, and polyacrylamide gels provide smaller holes, and that works well for smaller molecules. Okay? There's some other considerations to think about with respect to proteins. So if we want to separate proteins by a gel, that's a little loud, isn't it? If we want to separate proteins by a gel, there's a problem. With DNA, we don't have the problem. With DNA, we had a molecule that was a rod. Okay, that's the way we can think of DNA as like a rod, a long linear piece. And it's polyanionic, meaning its backbone is completely negatively charged. Every time we add on another nucleotide onto the size of a DNA, onto a DNA molecule, we add another negative charge. So the length is proportional to the charge. And that charge is fully negative. Well, what I hope you've been able to gather so far about proteins is that proteins don't have that property. Proteins at a neutral pH, for example, proteins may have a positive charge, they may have a negative charge, they may have a zero charge. So if I rely on the native charge of a protein as a means of separation, I'm going to have some very undesirable, unexpected things. Because if I put my negative electrode at the top, as I did in this gel that we can see here, if I put my negative electrode at the top, those proteins that are negatively charged will get pushed down. Those that are positively charged aren't going to be pushed down. They're going to come right back out. And the ones that are the most negatively charged may move faster than those that are less. Okay, so the charge to mass ratio doesn't make any sense for a protein. In a DNA molecule, the charge to mass ratio was a constant. Every time I added on another nucleotide, I added on another charge. So the length was proportional to the charge, and that provided me with a separation that varied only on the ability of these molecules to get through the matrix. If they're longer, they have more driving force going through. So the driving force per length of molecule is the same. The driving force per length of a DNA molecule is the same. Doesn't matter how long the DNA molecule itself is. With a protein, I don't have that. So with a protein, I've got to use some tricks to separate proteins by gel electrophoresis. Right? And one of the tricks that people use is very cool. Okay? It calls for adding a detergent to the proteins. A detergent. The detergent that's most commonly used is known as SDS. The name is sodium dodecyl sulfate. It'll be in your notes, but you don't need to know the name. But the acronym SDS you should definitely know. The types of gels that one uses to separate proteins are known as SDS-PAGE, P-A-G-E. The PAGE stands for polyacrylamide gel electrophoresis. So when you hear about SDS-PAGE, you know somebody is separating proteins, and they're using the detergent SDS as a way of doing that. Now, how does it work? How does that thing really work, right? Well, it's kind of cool. SDS is a negatively charged detergent. The sulfate part of the name is a negatively charged molecule. Because it's a detergent, it will denature proteins. How does a detergent denature proteins? Well, remember that proteins fold so that the inside of them is where the mostly hydrophobic residues reside. The detergent interferes with those interactions, and so what happens is the protein completely unfolds. So the reason you wash your hands before dinner is so you kill microbes by denaturing the proteins. Okay? Soap works just like detergent does. All right. Well, with SDS, when you add it to a mixture of proteins, all the proteins unfold, and each protein conveniently gets coated by this SDS. And it gets uniformly, for the most part, coated. Now, if the SDS is negatively charged, and it's uniformly coated, then that means that the longer the protein is, the more SDS will be on it, and the more negative charges will be on it, exactly like we had with the DNA molecule. The longer it was, the more negatively charged phosphates we had on it. And so we can use the same strategy then for separating the proteins as we used for separating the DNA fragments. And that's what we do. Okay? Now there's some other considerations about protein gels I'm not going to go into here, but I want to give you the basic idea, which is what I just described to you. Okay? So we're converting a protein from being globular into being more rod-like because we've denatured it, and it's coated with this negatively charged SDS. Well, if we put such a mixture of proteins onto a polyacrylamide gel, and we put the negative electrode at the top and the positive electrode at the bottom, we have a force pushing the negatively charged rods through this gel. And so what happens is the proteins separate, for the most part, on the basis of their size. There are some exceptions to that, but this is what a protein gel might look like. They've been separated on the basis of their size, and just like the DNA gel, the largest guys are at the top, and the smallest ones are at the bottom because they navigate through those holes the most easily. So SDS polyacrylamide gel electrophoresis turns out to be very, very useful. Okay, questions about that? Yes, question? How does the detergent interact with the negative portions of proteins? It doesn't have any problem interacting with any part of the protein, surprisingly. And it may be because when we look at a protein, it doesn't typically have a long stretch of negatives. Remember that when the protein is, I'm ringing here, when the protein is being coated by the SDS, the negative charges on the SDS are on the outside, not the inside, where they would interfere. Okay? And there are some exceptions. It's not a perfect system. Sometimes things don't exactly go according to their size, but usually they go pretty close to it. Yes? SDS stands for sodium dodecyl sulfate, and that'll be in the highlights. Okay? Yes, Deidre? I'd say 99.99% of the time people use SDS. Yeah. Back here? Oh, very good question. So this SDS, if I have it in here, can I pull it out of the gel? That's one question people have, and use it. And for the most part, no. Because the protein, once it's been denatured, usually is not going to come back together, or at least it's not going to come back together very efficiently. And so that's usually not a very viable option. Very good question. It's a very common question also. Okay. All right. So that is a cool technique. Here's sodium dodecyl sulfate. You can see that right there, SDS. And here's its structure. Long nonpolar chain. There's the sulfate, and there's the counterion of sodium that it comes as. Okay. Now, I want to talk about another method for separation, and it seems a little odd. It's called isoelectric focusing. I think you saw, you'll be seeing this week in your recitations. Okay? Or maybe you saw it last week. I can't remember when I assigned it. But in any event, what this method does is it provides a way of separating proteins by their charge. Okay? It separates them by their charge. And consequently, so how does it work? All right? Well, it works by starting with a bunch of little tiny beads again. And these little tiny beads are in a sort of a gelatinous mix. And the beads are of varying composition. Some of the beads have, let's say, 100 negative charges. Some have 99 negative charges. Some have 43 negative charges. Some of them have 21 negative charges. Some of them have 0 negative charges. Some of them have 15 positives, etc. So we can imagine a spectrum of these that go from, let's say, 100 positive charges over here to 100 negative charges, right? So we've got a whole spectrum of these molecules. If I take this mixture of all these little beads and I put them into a tube and I apply an electrical field to it, all right, the positively charged ones, okay, are going to move over here towards the negative electrode, and the negative ones are going to go over here. Well, I've got it backwards, but I've got the negative ones attracted to the positive and the positive attracted to the negative electrode. And so I get a whole gradient across this tube from very positive on the left side here to very negative on the right side, right? We can imagine right in the middle, that's where we would find the ones that are charged 0, okay? Well, that's a really nice way to set up the tube, but I want to separate proteins. Well, what I do is instead of just putting the beads inside of here, I put my protein mixture as well. So wherever the thing lines up with, let's say, -20, I will find proteins that have a charge of -20. So now I've separated these proteins on the basis of size. Well, these charges actually also correspond to the PIs, that is the pH at which the molecules are 0. It corresponds to the PIs of the proteins as well. So we have those that have a very low PI over here on the left, over those over here to the right were those that have a very high PI. So isoelectric focusing provides me a way of separating proteins on the basis of their charge/PI. Okay? Well, that turns out to be really useful, not by itself, but in conjunction with another technique I'm going to tell you about in just a minute. Okay? So separating on the basis of charge really turns out to be very, very useful. Okay. How does that work? Well, it works with a technique we call two-dimensional gel electrophoresis. 2 dimensions. Okay? How does two-dimensional gel electrophoresis work? Right? Well, as its 2 dimensions would suggest, this gel is run 2 times. All right? Let's imagine I start with a tube, and that tube has these little beads that I talked about that have the varying charges on them. And I take that tube and I add my protein mixture to this tube. I might, for example, take all of the proteins of a cell. Okay, this is very commonly done. Take cells, bust them open, and just take that whole mixture of all the proteins and put it into this tube. I apply my electrical field, and what's going to happen? Well, the proteins are going to separate on the basis of their charge, one side being very positive, one side being very negative. It doesn't really matter, but all right. And so now I've got a gradient of these proteins in this tube. Everybody envision that? Now let's take, I'm very careful, and I cut the tube open, and I take that gel that's sitting there, and I put it on top of an SDS-PAGE gel. You envision that? I add SDS, so my proteins that started out with a native configuration now denature in the SDS. They're still sitting in this little tube mixture. Everybody with me? Now I apply an electrical current, and I run it down through the SDS-PAGE gel. So first dimension, that is the first separation I did, was on the basis of their charge. And once I had them separate on the basis of their charge, now I separate them on the basis of their size. Okay? Everybody envision all that? If you can envision that, you're in great shape, because the results are remarkable, what you get with this. Okay? Here was that original tube. All right? So in this case, I show 4 proteins. That's very simple. When you typically wouldn't do it for 4, you would do it for thousands. Okay, but to keep it simple, we've got 4 proteins. They've been separated on the basis of their charge. The tube was cut open, SDS was added, and then were separated on the basis of their size. And what we see was what looked like 4 proteins actually contained multiple sizes there. These guys over here on the left, up at the top, we would look at that and say, okay, this red protein right here, was the one that was the most positive and the largest. This orange guy down at the bottom over here was the most negative and the smallest. Right? Everybody could interpret that reasonably well. Okay, so they're fairly straightforward to interpret. The beauty of this is when we actually look. Yeah, question?
Well, if the negative side is on this side, then what's the one?
The negatively charged proteins were on this side. So this is the most negative protein. Well, look, here's positive here. So this was the most positive protein. These were the most negative proteins. It's not the electrodes. We're talking about the proteins themselves. Okay? The electrodes would be opposite of that, yeah. Okay, so I've got small negative over here, right? Now, if I look at an actual gel from a 2D gel electrophoresis, this is what it looks like. Oh boy, right? We got garbage. Well, this garbage is really cool, as you're going to see. This particular plot shows all of the proteins of one cell type. You can look at this and say, well, based on that previous gel, the largest, most positive ones are over here. The smallest, most negative ones are over here. The largest, most negatives up here. The most neutral, middle-sized in here, et cetera, et cetera. Who cares? Well, I'll tell you who cares. The beauty of these types of gels is they're very reproducible. Okay? They're very reproducible. So if I run this same gel, these same proteins twice, I'm going to see the same plot. I can always look at this spot here where the plus sign is and say, oh, that's XYZ protein, because I can use techniques to identify what that protein is. And I can go over here and look at this big blob over here and say, that protein, I know what that is. The amount of darkness that I see for a protein corresponds to the amount of protein that I have. The more darkness, the more protein. Now, we haven't gotten to the really beautiful part of it yet. I'm just describing the isolation so far. Let's imagine now that I am a cancer researcher and I'm studying liver cancer. And I go and I have a patient who has liver cancer. And I want to understand what is it that's different about this patient who has liver cancer. I could take tumor cells from this patient and I could bust them open and get all the proteins from that patient's tumor cells in their liver. And I could go to the normal cells of that same patient, bust open those cells, and get all the proteins there. So I've got 2 mixes. One that is of proteins from the cancerous tissue, one that's from the normal tissue. I run 2 different 2D gel electrophoresis analyses, and I compare them. Because it's reproducible, I can look and say, whoa, look at this right here. This protein that I see right here doesn't appear in the cancer cell. Look, in the cancer cell, there's something great big that appears over here, and very intense, but it's not in the normal cell. I can completely profile the cancer cell versus the normal cell, and then I can say, now, which proteins are those? So instantly, I have a very good interpretation of how that cancer cell is different than the normal cell. There's other analyses. Let's imagine I've got a brand new drug, and I want to know the effect of this drug, okay, on cells. I could take one group of cells untreated, one group of cells treated, do the same analysis, and ask the question, where do the spots change? Right? So it's an extraordinarily powerful technique that allows me to analyze all of the proteins of a cell simultaneously. I can't do that in a one-dimensional gel. There's too many proteins, too many bands, and I can't see things. Here, I can separate every spot corresponds to a protein. Very cool stuff. Yes?
You know
How do I know this is not two things? It could be. It could be. Okay? So I'd have to do further analysis to determine that or not. It's possible that the reason it's smeared is because there's more than one thing there. Yep. How do you identify the specific proteins? Okay, that's going to come about in a minute, where I talk about a technique for determining the mass of proteins. Okay, so let me save this for that point. Okay. Everybody understand what we're getting here? Everybody get that? Okay. Pretty cool technique. All right. There's an analysis of colorectal versus normal, colorectal cancer versus normal. Okay. There are many examples of these you can look at and say, wow, there's really a big difference here. Maybe if I target this protein with a drug that inactivates the function of this protein, maybe I could have some effect on this cancer. And that's one of the things people do in developing drugs and thinking about how to treat cancer. Okay. So those are some pretty cool things. I spent a lot of time talking about purification. And so one of the things that we do with purification is we have to monitor it. We have to monitor. Remember, when in the very simplest sense, when we started our analysis, I said we busted open cells and we spun them down in a centrifuge, right? And when I spin things down in a centrifuge, I've automatically got two things. I've got supernatant and I've got precipitate, right? My compound, my protein, could be in either one of those or even both of those. So one of the things that people who work with proteins do is they monitor where is their protein. They don't want to throw the baby out with the bathwater. Oh, I don't need the supernatant. Goodbye. Oh, my God, there went my protein. Right? That happens a lot. Right? So monitoring what happens to the protein during the protein purification process, okay, monitoring that is important. This shows a set of steps that somebody went through in purifying a protein. And by the way, when you purify a protein, one step never does it. It takes multiple steps to get a protein pure. And during the process, you're monitoring it. Usually the protein has some kind of an activity that you can measure. Let's say an enzymatic activity. So you can say, okay, how much enzymatic activity do I see in the supernatant? How much enzymatic activity do I see in the precipitate? How much do I see in this peak that comes out off of the gel exclusion chromatography? How much do I see in the rest of it? That sort of thing. Okay? So I'm monitoring the activity of the protein. What you can see as this protein purification process went along, that the mixture got purer and purer. Well, that's what you really hope, because that's where the word purification comes from, is making this material purer. By the time they've gotten to the fifth step, they've got protein that's pretty darned pure. The monitoring of the process includes monitoring the activity, and that's actually shown in this table right here. Okay, so I'm going to step you through this table so you understand a little bit about what is going on. Here were the steps that we saw before, and I haven't talked about salt fractionation, so don't worry that you have to know that, okay? It's just a way of adding salt and making things precipitate is really all it is. Okay? This shows what we started with. We started with 15,000 milligrams of protein. We can measure the amount of protein in a mixture pretty easily. 15,000 milligrams. And we measured the activity of the protein that we were interested in there. And activity has just units. That might be amount of blue color produced or whatever activity that the protein has. We measured the number of units, and we found that it had 150,000 units. That meant that this protein had a specific this mixture had a specific activity of 10 units per milligram, because I had 150,000 units divided by 15,000 milligrams, 10 units per milligram. Okay? I had 100% yield because that was my starting material. I haven't lost anything. And that was my purification 1, meaning I haven't done anything except bust open the cells. So my starting point, there I am. After the second purification, I discovered that I've gotten rid of a lot of protein that I didn't want, as we'll see. Okay? So my total protein went from 15,000 milligrams down to 4,600. My activity went down. I lost a little bit of my protein, but I lost a heck of a lot more of things that weren't my protein. For example, I've lost about 3/4 of the mass of the protein that I started with, but I still have about 90% of the total units. meaning I've thrown away a lot of things that I didn't want, and a little bit of the stuff that I did want, that I wanted, went with that. But I'm willing to sacrifice that to purify my protein. That's what happens at every step, as you'll see. Okay? My specific activity went up. Units divided by milligrams gave me 30. Okay? And my yield was 92%. What does yield mean? Yield is the amount of activity that I have divided by the amount of activity I started with. Okay? So 138,000 divided by 150,000 times 100 gives me a yield of 92%. The last column is the purification level. And the purification level is simply how much purer it is than it started. How do I measure that? By dividing the specific activity that I have compared to what I started with. So 30 divided by 10 gives me a purification level of 3. The next step in the process, I go through ion exchange chromatography. Again, I get rid of a lot of protein. I lose a little bit of activity of my thing I'm after. But I can see that the specific activity went up, meaning I'm getting more and more of the protein is mine. That's units per milligram. Now it's up to 90. My yield is 77%, which again was 115,500 divided by 150,000, that which I started with. And my purification level is 9, meaning that my 90 was divided by 10 to give me that 9. Well, you can see then that each step in the purification process results in loss of some protein, but increasing amounts of specific activity, meaning that the protein is getting purer and purer and purer. At this point, by the time I get to the bottom, I have 30,000 units per milligram. I only started with 10. So when you talk about, hear about somebody improving the purity of something by 3,000-fold, that's what it means. It's 3,000 times purer than it was back here. Okay? So I've improved the purity of this protein by quite a ways. Questions about this? Is there a question? Oh, yeah. The purification will always be 1 on the first step. That's right. Because that's the amount of material that you start with. Yeah. How long do all these processes take? Boy, there's a lot of trial and error that goes in this. There's no rule. I mean, you could go through and do these. You might do these in, let's say, a week. You might have some technical problems along the way, and you might be doing this over a month. Okay? So it really varies. Okay, yeah, question? So purity versus yield, okay? So let's say that you get it more and more pure, but your yield is going down. So by the way, we're down to 35% of our total right here, which means that we've lost 2/3 of our protein. Is that good? Well, it is because it's a heck of a lot purer than it was, okay? And losing some of the protein that I'm after is a necessary evil of a purification process. And the reason that happens is because no purification process only gives one thing. So much as you would like to say, this purification process puts all of the molecules of my protein in this fraction and none of the molecules of my protein in this fraction, there's no purification method that's 100% efficient like that. So you're going to see that the yield is going to go down. And it's going to vary a lot from one purification method to another. This is not a bad purification level right here. Okay? Yes?
You're saying that for this particular protein, the purification level of 3,000 is reasonably good?
Yeah. Yeah, this is pretty good. Pretty good. Okay. Let's get to the question that somebody asked earlier about how do we identify those proteins? Okay? How do we identify those proteins? Before I tell you the specifics about that, we need to talk about one thing. And that one thing is that proteins themselves, when we go to analyze them, okay, The analysis that we're going to do on them works best not on complete full-length proteins, but better on small pieces of protein. Small pieces of protein are much easier to analyze than great big ones. We can separate them as great big ones. You've already seen that. But the analysis that we're going to do of those proteins is going to work best if we cut them up. Well, there are tools for cutting them up. There are chemical tools for cutting them, and there are enzymatic tools for cutting them. All right? So let's imagine I did that 2D gel. Okay? And I say, oh, wow, there's a spot right there. I'd like to know what that protein is. You can literally cut that spot out of the gel and get that protein out of the gel. You can actually munch it up, and you can have that protein that was on that spot in a tube. It's not difficult to do. Now, I've told you that that protein that you have in the tube, it's still intact. It hasn't been cut up in any way. It's going to be easiest to analyze if you break it up. So that's what I want to talk about first. Okay? And that is polypeptide cleavage agents. Okay? Polypeptide cleavage agents are enzymatic. That's what we're going to talk about most, and chemical. Let's talk about the chemical one first because it's very easy. If I take that polypeptide that I had in that spot that's now in my microfuge tube and I treat it with the chemical cyanogen bromide, what will happen is every place that there's a methionine, it will break that polypeptide chain on the carboxyl side of methionine. Remember that amino acids have carboxyl and amino sides. It's going to break it on the carboxyl side of a methionine every place there's a methionine. So let's say there were 4 methionines in there. I'm going to get 5 pieces. I break it 4 times, I get 5 pieces, unless methionines at the end. Okay? There are other chemical agents, and I won't make you know those. All right? My rule is if I don't talk about it, you don't have to know it, so I'm not going to talk about those. All right? Are those 4 things enough? Well, maybe they are, maybe they're not. I don't know where those are located in the protein. They might be very close to each other, in which case they're going to give me some odd sizes. So having other reagents that I can use to cut up the protein is helpful. One of these is an enzyme known as trypsin. So this is now an enzyme. An enzyme, of course, catalyzes a reaction. And the reaction that these enzymes catalyze is that they break down proteins by breaking peptide bonds. Trypsin specifically breaks peptide bonds on the carboxyl side of lysine and arginines. So I've got another tool now for making that big peptide into smaller fragments. Another common reagent that people use is chymotrypsin. Chymotrypsin cuts on the side of these side chains here. You see a common thing of these are all aromatic. And you see that these 2 here are fairly hydrophobic on the carboxyl side of those guys. Okay? Different ways of breaking up a protein. The last one I want to mention is this guy right here, carboxypeptidase A. It works pretty much by starting at the carboxyl end and chewing off 1 amino acid at a time. You chews them off 1 at a time. It notes here that not arginine, lysine, or proline, but we're going to treat it for simplicity's sake as if it cleaves every single one. Okay? Now, these have the effect of making a protein smaller. And the beauty of cyanogen bromide, trypsin, and chymotrypsin, oh, by the way, thrombin is also a reasonable, and thrombin is very easy, carboxyl side of arginine, is that they cut at specific places. They will give specific size fragments. So now I've got my proteins broken up into pieces, and I can analyze those pieces. And that's what we're getting ready to do. Any question about how we've broken them up? Yeah, back there. Would I like you to know the table? So what did I just say? Okay? Whatever I talk about. Right? So what I've talked about, yes, you're responsible for. Yes?
You're essentially saying with using each of these different chemical or enzymatic cleavages, you can divide proteins into size and analyze them and get the weight.
So, yes. So his question is, because I've got specific pieces, that means that it makes the analysis possible that I can do to determine what each one of those is. And the answer is yes. Yep. Okay, so the specificity of these enzymes turns out to be very helpful and useful, as we shall see. Okay, other questions? All right, so let's move to the analysis part. The analysis part is pretty cool. The analysis part is something that's developed largely in the last 15 years. There are several techniques for doing this. I'm going to talk about one. I'm going to jump down here to where it says MALDI-TOF. 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 MALDI-TOF is a means of analyzing peptides by mass spectrometry. Analyzing peptides by mass spectrometry. Mass spectrometry is a very powerful technique. It tells us the atomic mass of something, and it doesn't work very well on really large things. So its accuracy is improved the smaller the pieces are that it's analyzing. Okay? I'm going to tell you first how it works. Oop, that's not how it works. It works. Oh, blast it, I'm missing. I did this before. Okay, I'll get you a diagram that shows you MALDI-TOF, so I've got to describe it to you. How many people are familiar with what mass spectrometry is? Most people have had various chemistries, okay? So mass spectrometry works by having a detector, okay, that is able to analyze and tell masses that hit it. So when we do mass spectrometry, we have to have several things. First of all, it's done in a vacuum chamber. We have to have a way of ionizing molecules, because when we ionize the molecule, we use an electric field to let the molecule fly through the chamber and hit the detector. Okay? So for MALDI-TOF, we've got this mixture of peptide chains. And what we do is we embed it in a matrix. Matrix is just, you can think of, if you want to think of it as like a crystal of some sort or even a gel of some sort, that would be fine. So we've got our mixture of polypeptides embedded in this gel. We insert this mixture, which might be on the head of a pin, for example, into the vacuum chamber. Okay? We insert it into the vacuum chamber. So now we've got our sample, which has our peptides on it, in the vacuum chamber. And then we use a laser beam to hit the gel. Well, what happens to the gel when you do that is it vaporizes. And since the gel contained our peptides, they vaporize as well. Now, two cool things happen with that. During the process of vaporizing, many of the fragments that were in that gel acquire a positive charge. They acquire a proton. They're now positively charged. If I apply an electrical field, what's going to happen? Well, if I put negative down here and positive over here and it ionizes right here, what's going to happen? Boing, it's going to go towards the negative field. The time it takes to move from the place where it ionized to the field is going to be a function of its charge and its mass. If I put one charge on there, let's imagine I only put one charge on there, and I've got a mixture of compounds here. One of them is at, let's say, 1,000 molecular weight, and another one is at 2,000 molecular weight. They both have a positive 1 charge. Which one's going to move faster? The 1,000, right? because its mass-to-charge ratio is smaller. The force that's moving it is the charge. So if I have the same force, it's kind of like saying, here, go push this car that's out of gas. Do you want to push a Prius, or do you want to push an SUV? Well, you're probably going to push a Prius faster than you're going to push an SUV, right? Because it doesn't have as much mass. You have the same amount of force. It takes less time. It takes, in fact, half the time for the 1,000 molecular weight compound to get there as it does the 2,000. All right? What if I had the 2,000 molecular weight compound that had 2 charges and the 1,000 molecular weight compound that had 1 charge? They would arrive at the same time, right? The mass-to-charge ratio is equal. Generally, these guys don't get very many charges. They usually get 1. They may sometimes get 2. But we can sort those out. Well, the beauty of this is we can tell now the masses of all the things that were in there. We can tell the masses. We can look at that and say this thing had a mass of 987.31 grams per mole. And we know the masses of all the amino acids. We could ask the question, we could ask the computer to answer the question, which amino acids, when put together, give 978.83 grams per mole? We have a way of identifying those fragments. Something that helps us to identify those fragments is something else that happens in that chamber. When I put that positive charge onto the fragment, to a limited extent, breaks apart. That might sound like it complicates things, but if you think about it, it's breaking apart into amino acids that we know the mass of. We know what they are by their mass as well. So, for example, if I were to look at, where am I at here? This guy, here would be a fragment I might have in that chamber, and here are various places where this fragment might break. Here's the mass of the full one. Here's the mass of this without the glutamic acid. Here's the mass of it without the glutamic acid. Well, 2 glutamic acids, I guess. Okay. Each time I'm breaking off a specific piece, and I can look at the difference between the masses of these and say the difference between this and this is, oh, that was a glutamic acid. The difference between this and this was another glutamic acid. The difference between this and this was a glycine. And so what I'm doing by reading these different masses off is I'm identifying for sure what the sequence was of those amino acids in that peptide. Now this is really, really powerful because I usually know the sequence of every protein in a cell because I've done genomic sequencing. I now ask the computer, which protein has the sequence glu, gly, met, arg? I'm going to get a very small subset of all the total proteins. I put these all together, I can eventually identify which protein corresponded to that set of fragments that I had. Okay, question over here.
Because of the different side chains, each amino acid it has a unique mass then?
Yes. There's no confusion based on the amino acid because they each have unique side chains. That's correct. Okay? Pretty cool. This technique is remarkable. Okay? You could take that 2D gel that I described to you earlier. You can take the 2D gel, and there are robotic machines that will do the work that I just described. That is, they will go, they will cut out the spot, they will extract the protein from that spot, they will digest it with enzymes or chemical cleavage agents, they will put it into the mass spectrometer, they will get the spectra, which is what you see here, they will analyze that, and they will tell you every spot that was on that gel. These machines that are out there can do 4,000 proteins a day. Okay? Now, to tell you how old I am, when I was doing my master's thesis back in the long time ago, all right, I did a lot of mass spectrometry, all right? I did a lot of mass spectrometry. And if we could do the analysis of one molecule in a week, we were delighted. And that one molecule in a week wouldn't be anything nearly as large as this. Okay? To do 4,000 in a day is remarkable. It's really remarkable. So this technique is very powerfully used in a field we call proteomics. Proteomics. I'm going to define that for you. Proteomics is the study of all the proteins of a given cell. Proteomics is the study of all the proteins of a given cell. You've heard of genomics, probably. Genomics is the study of all the DNA of a cell. When people did genomic sequencing, that is, determine the sequence of DNAs of cells, they were doing genomics. When they're doing this kind of an analysis, they're doing proteomics. And the 2D gel electrophoresis is part of that, because it allowed that separation. Questions about that? This doesn't tell you anything about quaternary, no. Because remember, you're denaturing the proteins to start with, right? This is going to tell you what the sequence of any given spot is. Okay, so I've got two things for you. One is I forgot to give you the limerick of the day. Let's go get the limerick of the day. Oh, yeah. The limerick of the day is, whenever there is a snowstorm, the cattle all seem to conform. In actions I've found by hugging the ground as a way to keep each other warm. Ah, ha, ha. Bad, okay. The song of the day was actually written by one of my former students. She was a TA here, as she's presently in the PhD program at Harvard. She's a really remarkable young woman. And her song is entitled, "The Proteins Marching One by One." It's to the tune of "The Ants Go Marching One by One." Oh, there's a method you should know that's very huge. It's spinning round and round inside the centrifuge. The supernatant pellet too, you choose the one that's right for you, and from there we purify what's inside. To size exclude filtration is the way to go. The beads have pores where molecules go in, you know. The largest ones eluding fast, the smallest ones will come out last, and the proteins purified by their size. Electrons power gel electrophoresis. The protein is denatured thanks to SDS. Proteins in a minus state get sorted by atomic weight. Smaller ones in speedy mode to the anode. Ion exchange is special chromatography. To switch cations, you must have a minus speed. Upon the speed, the proteins bind. They're positive, not any kind, and the others wash right through. Out to you. Oh my, this song has given you a mighty list. Perhaps we'll just skip over old dialysis. So study HPL and C. If you have questions, talk to me. You will get through protein hell. You'll do well. All right. See you guys on Wednesday.